Here's active and passive transport for the MCAT in 2 minutes. We're looking specifically about how it's tested, so make sure you understand it like this. And check out my MCAT community where you can find hundreds of PDFs with pictures like these and typed out explanations. Pumps and channels are two of the main ways that we move molecules in and out of a cell. Pumps are always active transport, and active transport always means that molecules are moving against their concentration gradients. This pump here is moving potassium from the outside of the cell to the inside of the cell, so that means that the inside of the cell has a higher concentration of potassium than the outside of the cell. And the opposite is true of the sodium. The sodium is moving to the outside of the cell, so that means that the outside of the cell has a higher concentration of sodium than the inside of the cell. Make sure you understand the importance that if they use the word pumps, that means it's active transport. It can never be passive transport if they say a pump is being used. We'll talk more about this in a second, but anytime you're moving something against its concentration gradient, it has a positive delta G and is non-spontaneous. Channels are the exact opposite, and they move things with their concentration gradient. So for this glucose molecule here, it's moving from a high concentration of glucose to a lower concentration of glucose inside the cell. This type of transport is called passive transport. And once again, if they use the word channels, that means that it has to be passive transport, and that molecule is moving from a high concentration to a low concentration. All passive transport has a negative delta G and is spontaneous. They like to test your understanding of the energy considerations for these, so we'll look more at that right now. We said active transport would have a positive delta G, so this graph here is what's representing that. We're moving molecules from a low concentration, so that's basically your reactant, to a high concentration, which we can say is the product. Since this has a positive delta G, we have to pair it with a reaction that has a negative delta G, and very often that reaction is the hydrolysis of ATP. The energy released from that hydrolysis that we can see in that graph is then used to move those molecules against their concentration gradient. However, this part doesn't always have to be the hydrolysis of ATP. There are two really common examples that you need to be aware of that don't use ATP. One of them is in the ETC whenever we are transferring electrons. That transfer of electrons is a spontaneous reaction and it has a negative delta G, and the energy released is what's used to drive the protons from the matrix of the mitochondria to the intermembrane space, because the intermembrane space has a higher concentration of protons, so we have to use active transport to do that. And the other common source of energy is passive transport. Remember how we said passive transport has a negative delta G, and that's pictured right here. You can think of the reactants as the molecules at their high concentration, and then the products as those same molecules now at their low concentration. When they move from high to low, they release that energy. That energy can then be used to pump other molecules against their concentration.